Hi everyone, welcome to this tutorial on the molecular mechanisms of action potential generation. So in the previous few sessions we've been talking about the ionic basis of action potential generation. Now we want to move more towards the biological question of what are the mechanisms that are responsible for the production of this signature electrical event that we find in excitable tissue. All of this relates to one of our neuroscience core concepts, the one we've been focusing on, the fact that neurons communicate using both electrical and chemical signals. So we continue to talk about the electrical signaling activity of neurons, and very soon we'll be moving in to talk about the chemical signals. Well, for today's session, we have some learning objectives. I want you to be able to describe the molecular properties of sodium and potassium channels and understand and recognize how these properties explain the voltage and time-dependent permeability changes that underlie the generation of action potentials. And secondly, I want you to be able to describe the molecular mechanisms that establish the chemical gradients for these important ions, sodium and potassium, across the neuronal plasma membrane. Well, to begin, let's again reiterate some of our foundational principles. That is that changes in potassium and sodium permeability underlie the action potential. So we've seen this figure a few times before. It allows us to conceptually understand the resting membrane potential as being a state dominated by the leak conductance for potassium, the rising phase being a phase of action potential generation where there's an explosive increase in the permeability of the membrane for sodium, such that at the peak of the action potential, the membrane potential comes very close to the Nernst equilibrium potential for sodium, because the sodium permeability of the membrane is so much greater than any other ionic permeability. The ability to predict membrane potential becomes, as a first approximation, the Nernst equilibrium equation for sodium. Well, the falling phase of the action potential involves a dramatic reduction of sodium permeability, and this is the inactivation of this sodium conductance. We'll talk about the molecular mechanisms for that inactivation in this tutorial. But in addition, we know that the voltage and time dependency of the potassium conductance means that the potassium permeability is rising at the same time that sodium permeability is collapsing. And those two factors together allow us to return to our resting condition, where once again the permeability of the membrane, due to the fact that there's some leakiness of the membrane to potassium, is greater than sodium permeability. And once again the resting membrane potential is established, being close but not precisely the Nernst equilibrium potential for potassium. Well, this conceptualization allows us to again understand how changes in membrane permeability lead to changes in membrane potential, and as we discussed last time, and we'll talk about the mechanisms today, changes in membrane potential can in turn have an impact on changes in membrane permeability. Now, I would encourage you to view again the animation that's available on the website that supports our textbook. It's animation 2.3 on the action potential. You can click following the hyperlink on your tutorial notes, or just navigate on your own to that website. All right. Well, for today, what we really want to understand is what are the molecular mechanisms that allow a change in membrane potential to impact membrane permeability. And this drives us to consider the actual integral membrane proteins themselves that constitute ion channels for sodium and potassium. We'll do so in two ways. First, we'll consider a functional model for how this feature, a change in membrane potential, leads to a change in membrane permeability. And then we'll consider the actual molecular structure of these proteins and gain some insight as to how this plays out in actual biological terms. So first, a functional model. Now, This is an experiment that should be familiar to you now. What we're doing is using our voltage clamp method to apply a step depolarization that is going to be supra-threshold, that is well above threshold for the generation of action potentials. And rather than showing you the action potential or the voltage and time-dependent changes in conductance, I want to show you our current understanding of what's happening at the level of the integral membrane proteins that constitute permeability channels for sodium and potassium. So let's step through this figure in time, and let's look at what happens at the onset of depolarization. So that would be time 0 when we first depolarize. Now, recall that the inward current is fast. That is, the increase in sodium conductance is quite rapid relative to the increase in potassium conductance. And now we gain some insight as to how that might happen. Almost instantaneously, with the depolarization step, the probability of sodium channels reconfiguring from their closed state to the open state is dramatically increased. In other words, sodium channels open at the onset of depolarization. Now notice what's going on for potassium channels. The probability that a potassium channel will go from a closed state to an open state is less than for the sodium channel with this step in depolarization. If we consider the behavior of a single ion channel, which it's possible now to do, we can understand this behavior in more principal terms. But on the aggregate, what we can say is that the sodium channels respond as a collection of proteins more rapidly with greater probability of opening with the onset of depolarization relative to the potassium channel. This is why the positive feedback phenomenon is so rapid, and why the repolarization phenomenon lags the explosive rising phase of the action potential. Okay, so let's move a little bit further out in time. Now we are a few milliseconds later, and we see that the sodium conductance is beginning to inactivate. Why? Not because the pore of the channel is pinching down, but because there is a particular mechanism at play here on the inner aspect, that is the cytoplasmic surface of the sodium channel. There is a gate that we call the inactivation gate that swings shut as the membrane becomes depolarized. So we'll talk about what exactly that is and why that's happening in just a minute. But for now, I want you to note that inactivation of the sodium channel is due to the fact that there is this particular cytoplasmic feature of the channel protein that swings closed and plugs up the pore of the membrane from the cytoplasmic side, preventing any further influx of sodium channels. And this is why that sodium conductance rises so rapidly and then falls equally rapidly, because of the development of inactivation. Now, at about this time, our potassium channels are opening up more consistently. That is, the probability of channel opening is greater, and more potassium channels are in fact in the open configuration, allowing for potassium to flow out of the neuron, this contributing to repolarization and eventually the undershoot of membrane potential. Now notice the cytoplasmic surface of this potassium channel. There is no inactivation gate, at least on this particular type of potassium channel. So these channels are going to continue to remain in their open configuration, provided that we continue to depolarize this membrane, in this case with the voltage clamp experiment. It's only with the cessation of depolarization, that is, when we step back down near the resting membrane potential, that we induce a change in both kinds of channels. This hyperpolarization is essential to restore the closed state for both of these channels. For the sodium channel, that means that the pore itself is closed and the inactivation gate is open. For the potassium channel, it means that the pore simply has closed off, preventing any further efflux of potassium ions. Now let me again restate the point about the sodium channel because it is important. Notice that there's a difference between inactivation and closed. In the inactive state, the pore of the channel is open, but the gate has closed on the cytoplasmic surface of the channel. In the closed state, the pore is stopped up, preventing further flow of ion. However, the inactivation gate has been reset and restored. So when the channel for sodium is back to its closed state, we are now outside of the absolute refractory period. It's possible for this channel to open up again. That is, it's possible for this channel to re-enter a cycle that will allow for channel opening and the influx of sodium. So the absolute refractory period of the action potential is defined in terms of the molecular structure of the ion channel itself. When these channels are inactive, they are incapable of passing current. That inactivation is only relieved with hyperpolarization. So this inactivated state is when we have our absolute refractory period. Okay? All right. Well, the relative refractory period then is a time following sufficient hyperpolarization to reset that sodium inactivation gate, but it's a time when these potassium channels may remain open and we may still be in the undershoot of the action potential, so it will require a stronger stimulus to activate that sodium channel sufficiently to induce that positive feedback circuit that leads to this explosive regenerative rising phase of the action potential. Okay, now let's move on from this functional consideration of how sodium and potassium channels work relative to their voltage and time dependencies to consider the actual molecules themselves. Well, these ion channels are very complex, but they do have some features in common, and I want to emphasize their common features and just highlight some of the differences. So let's begin by thinking about the sodium channel. Well, the sodium channel that we've been talking about is really a complicated integral membrane protein that contains multiple motifs in which there are helical segments of the protein that are passing through the membrane. So we can recognize 4 motifs, with each motif being comprised of 6 transmembrane segments. And these motifs fold together in a 3-dimensional shape within the membrane, constituting the voltage-gated ion channel that's selectively permeable for sodium. Now, as this protein establishes its tertiary shape in the membrane, it's configured in such a way to establish a pore for the passage of sodium ions. And the electrical and physical structure of that ion channel pore account for its selectivity for sodium. I'll also highlight these yellow transmembrane segments that we see in each of these motifs. And these contain positively charged amino acid residues that account for the voltage sensitivity of this protein. Now, there are also additional components to this channel. There's something called a beta subunit that aggregates with the main integral membrane protein that forms this channel. So this is the sodium channel. We think there are probably at least 10 sodium channel genes that express the proteins that are essential for building these channels. But those genes can have multiple reading frames. and the product of that gene can be modified in various ways. So there may be different capacities to modify this basic structure in interesting ways that will have an impact on its function. And in some cases, there may be genetic mutations of these genes that will impair the function of these channels. And that's a fascinating story that you can read more about in a box that's found in Chapter 4. Okay, so that's a brief look at the sodium channel. We actually know more currently about the molecular structure of potassium channels. One perhaps surprising fact is that there are a large number of genes that encode potassium channels, and potassium channels can have multiple different forms that have different physiological properties owing to their distinct molecular structure. The ion channels that are constituted from these gene products typically represent the aggregate of multiple subunits. That is, not just one integral membrane protein, but more commonly 4 integral membrane proteins that come together and constitute the voltage-gated potassium channel, or potassium channel that's gated by some other mechanism. There's a large family of these channel genes and a large number of variations on an ion channel that allows the passage of potassium. So far, we've been talking about the kind of voltage-dependent ion channel that's represented over here on the left-hand side of this illustration. This is a channel that is gated by a change in voltage. So it, like the sodium channel, has a voltage sensor. We'll talk more about that in just a moment. But there are other kinds of ion channels that are sensitive to other factors, such as calcium within the cell. And that is attributable to a cytoplasmic portion of this integral membrane protein that can interact with calcium. And there's a large variety of other potassium channels that we won't mention at this point, but together they contribute to the exquisite regulation of excitability and the nature of electrical signals that neurons can generate. Now, let's back away just a little bit from the more detailed look at the molecular structure of the voltage-gated ion channel and consider, again from a more functional perspective, how these various components aggregate together to form a functional ion channel. So this is a model now of a voltage-gated potassium channel. So we imagine that these 4 subunits come together to constitute a channel, and there's something that we'll call a pore loop that forms from the aggregate of these 4 subunits. And these pore loops together establish the ionic selectivity and the size selectivity for the passage of potassium ions. Now, we've already talked some about the voltage sensor that's part of each subunit, and that voltage sensor is illustrated by these wings out to the side of these subunits that carry positively charged amino acid residues. So in the hyperpolarized state, when the inside of the cell is made more negative, these positive charges in these wing-like structures are drawn inward toward that negativity. The conformational change that happens as these subunits pivot allows for a closure of the pore loop regions, and potassium ions are not able to pass outward through this channel with hyperpolarization. Now imagine what will happen if we depolarize the cell, that is, we make the inside of the cell positive, perhaps by the opening of a voltage-gated sodium channel. Now these wings that bear these positive charges are pushed away by electrostatic forces, and that pivots the subunits in a way that now opens up a pore in the center of this channel, and potassium ions are then able to aggregate and pass out of this pore region. So the function of these ion channels is a reflection of their structure, and the voltage dependency and even the time dependency has to do with the structure of these voltage sensors and perhaps the density of charged residues that might contribute to the kinetics of channel opening. Now, as I mentioned, one of the consequences of the genetic encoding for these ion channels is that mutations can occur that have consequences for neurological function and, in some cases, human health. And you can read more about that topic if you're interested in Box D in Chapter 4 of our textbook.